There's something in the air tonight that wasn't there 3 billion years ago. Well, the air itself wasn't there, really. Not the way it is now. What existed instead was a sky heavy with carbon dioxide and methane and nitrogen. A sky that would have been lethal to nearly every organism alive on this planet at this moment, including you. Especially you. It's a strange thing to sit with, that the atmosphere pressing against your skin right now, thin, forgettable, invisible, was made. Not all at once, not by anything with intention, but made nonetheless, by organisms that had no idea what they were doing, which is, when you think about it slowly, the way most large things happen. Something this warm probably exists somewhere in your house tonight, a bowl left out, the damp underside of a rug near a door. You don't think of those spaces as biologically active, but they are. They have always been. Welcome to the Sleepy Biologist. If you've been here before, you know the pace. If you're new, there's nothing to keep up with. This channel keeps going because of the people who quietly support it, who press the like button before they fall asleep, who've turned membership into something more like a habit than a transaction. That matters more than it probably looks like it does. The organisms we're talking about are cyanobacteria, and the name matters less than the fact of them. They appeared somewhere around 2.7 billion years ago, possibly earlier. The fossil record gets uncertain at those depths, and it's worth saying that plainly, we are working from compressed rock and chemical signatures and a lot of inference. What we have is good. What we have is not everything. What we know is this. Cyanobacteria develop the ability to use sunlight to split water molecules. And when you split water, H2O, you get hydrogen and oxygen. The hydrogen gets used, the oxygen gets released. At first, the oxygen was absorbed almost immediately by dissolved iron in the oceans. There was a lot of iron. It took hundreds of millions of years before oxygen started accumulating meaningfully in the atmosphere. The planet was consuming what these bacteria produced as fast as they could produce it. A slow, unglamorous standoff between a gas and an ocean. Then the iron ran out. Oxygen started building up, and the organisms that had been living perfectly well in an oxygen-free world began dying. It was, depending on how you frame it, the largest extinction event in Earth's history, or just chemistry, the Great Oxidation Event, somewhere between 2.4 and 2.1 billion years ago. A catastrophe and a clearing happening at the same geological moment. The thing that I keep returning to, and I'm not sure this ever fully resolves, is that the organisms responsible for this had no concept of consequence. There was no moment of decision. Photosynthesis was simply what they did, the most efficient way to extract energy from their environment. What followed was incidental to their survival in a way that is almost uncomfortable to describe. They remade the world while trying to eat. Cyanobacteria are still here. That's the part that sits quietly under everything else. They're in the soil beneath your nearest park. They're on building facades, in the crusted biological skin that forms over desert surfaces, in rice paddies, in Antarctic melt pools. They're in the ocean in numbers that don't pass easily. Somewhere in the range of 10 to the power of 24 individual cells, a 10 with 24 zeros behind it. That number is physically hard to hold in the mind. It was for me when I first tried. They still photosynthesize, they still release oxygen. An estimated 20 to 30% of the world's oxygen supply comes from cyanobacteria specifically, not from trees, not from the rainforest as a whole, from organisms you cannot see without equipment, in environments you might not think to look. A breath you take tonight will contain oxygen that cyanobacteria released, possibly within the last few months, possibly from a bloom in a lake you've never heard of, possibly from the surface of soil 2 miles from wherever you're lying right now. There's something worth slowing down inside if you can. The sensation of breathing, the slight expansion across your chest, the pause before exhale, is built on a biological infrastructure that predates every complex organism by billions of years. Your lungs are recent, extraordinarily recent. The oxygen moving through them is not. The lineage of that gas is longer than animals, longer than plants, longer than fungi. It arrives in you at the end of a chain of events that began before the Earth had a moon in its current orbit, before multicellular life existed in any form, before the oceans were their current salinity. You breathe at the end of that. There's a word, stromatolite, for the layered rock formations that cyanobacteria built up over millions of years. They're photosynthetic mats slowly mineralizing into structures you can still find in certain shallow bays in Australia. They look unremarkable, flat, rounded, dull brown at the surface. They're also some of the oldest direct biological records on the planet. You can stand in front of one, and what you're looking at is the closest visible remnant of the organisms that first oxidized this atmosphere. There's no museum label that quite earns what that means. I've seen photographs and felt faintly that the photographs don't entirely work, that some things resist being captured in a format you can scroll past. The metabolism of a cyanobacterium is not complicated in the way animal metabolism is complicated. In broad terms, and the details are biochemical enough to become a different kind of narration, what happens is this. Light energy is absorbed by pigment molecules used to extract electrons from water, and those electrons are passed through a chain of proteins that ultimately powers the construction of sugar from carbon dioxide. Oxygen leaves as a byproduct. Sugar stays. The proteins involved, photosystem 1 and photosystem 2, though the numbering is counterintuitive because 2 actually runs first, are among the most studied molecules in biology. Researchers have been examining them for decades, and there are still structural questions, still slight uncertainties in how the water-splitting step works at the atomic level. That's not a failure of science. It's just the texture of how knowledge accumulates in a field where the thing you're studying is smaller than a wavelength of visible light. At night, and you are in night right now or something close to it, cyanobacteria don't photosynthesize. Without light, the machinery pauses. What continues is respiration, the same basic process of breaking down sugars for energy that runs in your cells. In the dark, cyanobacteria and humans converge on the same ancient biochemistry. Different shapes, same core chemistry. It's a strange family resemblance. Some cyanobacteria, the ones in extreme environments, reduce their metabolic rate to almost nothing under prolonged darkness. They wait, not consciously, not in any way that involves patience as a felt experience. But they persist in a kind of minimal state that lasts until light returns. There's a quietness to that which doesn't quite have a name. Your body, for all the distance it has travelled from that ancient chemistry, still runs something recognisably similar at its cellular core. The mitochondria in your cells, the ones generating energy right now as you read or listen or begin to drift, are the descendants of bacteria, not metaphorically, structurally, genetically. About 1.5 billion years ago, a bacterium was engulfed by another cell and not digested. It stayed. It reproduced inside its host. Over generations, it became integrated to the point where its descendants can no longer survive outside the cells they inhabit. They kept their own small genome. They still have it. It's much reduced from what the original bacterium carried, but it is there. Inside you right now are the reduced remnants of an ancient independent organism keeping your cells powered. The fact that this is commonplace knowledge doesn't flatten it. it still takes a moment to actually mean something. Somewhere beneath a forest floor, or the soil at the edge of a road, or the surface of a rock face, or the shallow margins of a pond, cyanobacteria are doing in darkness what they've always done in darkness, waiting, metabolizing slowly, surviving the night with a patience that predates the word patience, that predates anything that could experience patience as a state. And when light comes, whenever light comes, they will begin again. It is not remarkable to them, it is not anything to them, but you are here in part because they never stopped. Deeper in the soil, beyond the light-dependent surface, something else is underway. Organisms that never learn to photosynthesize, that have only ever extracted energy from chemical reactions in the dark. They are working through the night in ways that have almost nothing to do with oxygen or sunlight, or anything the surface layer needs. Their chemistry is older in some ways, more brutal, and it powers a different part of the planet's invisible engine. There's a smell I've never fully gotten over. Salt marsh at low tide on a grey afternoon in October. The kind of day where the sky and the water are almost the same colour and you can't tell where one ends. I was there for a field assignment, crouched in muck up to mid-shin, and the smell was, there's no clean way to say this, sulphurous, heavy, the kind of smell that registers as wrongness before your brain can name it. My instinct was to step back. My field supervisor said, without looking up from her sample jar, "That's what productivity smells like." I was briefly offended. Then I spent 2 hours reading about it. Then I became someone who finds that smell interesting, which is its own kind of loss. What I had been smelling was dimethyl sulfide, a gas released when sulfur-reducing bacteria break down organic matter in low-oxygen sediment. And the bacteria doing that weren't decaying the marsh. They were running it, processing it, moving material through a cycle that the marsh depended on. The smell was a byproduct of work. Enormous, continuous, invisible work. That reframe still lives in me somewhere. It changed how I think about the word decomposition. Here is what decomposition is not. It is not things falling apart. That framing is almost completely wrong, and I held it for longer than I'd like to admit. What decomposition is, the actual biology of it, is a transfer. A careful, layered, slow redistribution of material from one form into others. Nothing is lost. Everything is changed hands. A leaf falls, you've seen this. You know the sound it makes when it lands, papery, light, almost nothing, and then it just sits there. It begins to look less like a leaf. The edges soften, the colour drains. Eventually it becomes indistinct from the surface it landed on. What is actually happening in that interval, the weeks, the months, sometimes longer, involves so many organisms working in such different registers that trying to describe them sequentially starts to feel like a lie. They're not sequential, they overlap. They depend on each other in ways that aren't always obvious or even fully mapped. But roughly, and roughly has to be good enough here, it starts with water. Moisture moves into the leaf. Cell walls that were once maintained by living pressure begin to lose their structure. Fungi arrive early, often before bacteria in significant numbers, because fungal hyphae can penetrate intact plant tissue in ways that many bacteria cannot. They're probing through, extending into the leaf's interior, secreting enzymes that break down cellulose and lignin, the fibrous components that gave the leaf its shape when it was alive. Lignin in particular is stubborn. It's one of the more chemically complex polymers in plant biology, and most organisms can't touch it. The ones that can, certain fungi, certain bacteria, are genuinely essential to carbon cycling in forests. Without them, dead plant matter would just pile up. Carbon would accumulate in structural plant tissue and stop moving. The whole cycle would grind towards something much slower and less functional than what we have. White rot fungi, which despite the name can be various colours, are among the most effective lignin degraders on the planet. There's something I find quietly satisfying about that. The most chemically intractable material in a forest being handled by an organism with no brain, no circulatory system, nothing that resembles intention, just enzymes, just chemistry, running continuously. The bacteria arrive in waves, or something like waves. I'm not sure that word is precise enough, but the alternatives are worse. Different species become dominant as the chemical composition of the leaf changes. Early decomposers create conditions for later decomposers. The breakdown products of one organism become the food source of another. It's not cooperation exactly. No one decided anything, but the outcome is cooperative in effect. Soil invertebrates move through, springtails, mites, millipedes, earthworms in the lower layers. They fragment the material, physically breaking it into smaller pieces, which dramatically increases the surface area available to microbial action. An earthworm doesn't digest the way you do, really. It passes material through its gut, and what comes out is transformed. pH shifted, microbial communities altered, nutrients more available than they were when the material went in. The castings, which is the polite word, are among the most biologically active material in a healthy soil. I spent a semester turning compost twice a week in a university garden, and I want to say, because it took me a while to really feel it, that the moment a compost heap heats up from the inside is not a small thing to witness. You put your hand into the centre of a pile and it's warm, not from the sun, from organisms, from metabolic activity. Billions of bacteria generating heat as a byproduct of breaking down organic material, the same way your muscles generate heat when they're working. Invisible labour made tangible by temperature. It was one of the more quietly astonishing things I have witnessed in a field full of astonishing things, and I have never entirely stopped thinking about it. The carbon in a decomposing leaf doesn't disappear. Some of it is incorporated into microbial biomass, the bodies of the organisms doing the decomposing. Some is released as carbon dioxide, returned to the atmosphere, made available again for photosynthesis. Some is processed into humus, stable organic compounds that can persist in soil for decades or centuries, holding moisture, providing structure, making the difference between soil that grows things and dirt that doesn't. And the nitrogen. The nitrogen in that leaf, fixed originally by bacteria somewhere in the soil, moved through roots and through the plant's own chemistry into the leaf tissue over a growing season. The nitrogen gets recycled too. Broken down from organic compounds into ammonium, then nitrified into nitrate by specific bacterial groups, made available again for plant roots to take up. The same nitrogen that was in the leaf may be in a root tip by next spring. It may be in whatever grows from that root by summer. There is a category of thinking I'm suspicious of, which is the idea that decomposition is a secondary process, something that happens after the real biology, the living and growing and reproducing, is finished. The clean-up crew, one of my professors used to say, and I remember finding that framing comfortable at the time. It's not the clean-up crew. Decomposition is not after life. It's part of the same cycle, running at a different speed. The organisms doing it are not cleaning up after the living. They are living. They are metabolizing, reproducing, competing with each other, dying, being decomposed themselves. Every gram of healthy topsoil holds somewhere between 100 million and 1 billion bacterial cells. I've run the assays. The numbers don't feel real when you first see them. They feel like a decimal error. They're not. The leaf you're imagining, the one that fell, the one softening at the edges, is not decaying. It's being taken apart by things that need it, things that have evolved across geological spans of time specifically to process what living organisms leave behind. The relationship is old, older than forests. There were decomposers before, there were complex things to decompose. They were ready. What they make from that leaf is not nothing, it's the next thing. The nitrogen in your body was not always yours. That's not a poetic way of saying something, it's just the literal situation. The nitrogen currently in your muscle tissue, in the DNA coiled inside your cells, in the proteins your body spent energy assembling, it came from somewhere. It passed through things. And before it was in any of those things, it spent an incomprehensible stretch of time as N2, a molecule so stable, so stubbornly unreactive that almost nothing in the biological world can touch it. Almost nothing. Nitrogen makes up about 78% of the atmosphere. It's the dominant gas in every breath you take, and your body ignores it completely. You inhale it. You exhale it. Nothing happens. The molecule passes through you like you're not there. Because to nitrogen gas, in a biological sense, you aren't. You have no mechanism to use it. Neither do plants. Neither do most organisms. This is one of those facts that I sat with for longer than it probably warranted during grad school because the implications are genuinely strange. The element that makes up a substantial portion of every protein you've ever built, every strand of nucleic acid in every cell of your body, that element is everywhere. It is literally in the air, and virtually all of it is inaccessible to you. The organisms that bridge that gap are bacteria, a specific group doing a specific thing that the rest of the living world depends on absolutely. Nitrogen fixation, the conversion of N2 into ammonia, NH3, requires breaking a triple bond. One of the strongest bonds in chemistry. The enzyme that does this, nitrogenase, is a strange and somewhat delicate piece of biological machinery, sensitive to oxygen in a way that creates obvious problems for organisms that also need oxygen to survive. Some nitrogen-fixing bacteria deal with this by fixing nitrogen only at night. Others sequester the enzyme in specialized cells. Others live in environments with very low oxygen concentrations and don't have the problem to begin with. The metabolic cost is steep. Fixing one molecule of nitrogen requires somewhere around 16 ATP, the cell's energy currency, depending on conditions. That's an enormous expenditure for a single molecular conversion. The fact that organisms do this at all, that they evolved and maintain such expensive machinery, is some indication of how badly the living world needed it. Cyanobacteria do it. Free-living soil bacteria like Azotobacter do it. But the system most people encounter, in the rare moments anyone thinks about this, is the symbiosis between Rhizobium bacteria and legume roots. The legume detects chemical signals from Rhizobium in the soil. The bacteria detect signals from the plant. There's a slow negotiation, flavonoids released by the root, nod factors released by the bacteria in response, root hairs curling, bacteria entering through an infection thread, the root forming a nodule around them. The nodule is a purpose-built structure. The plant constructs it specifically to house these bacteria and maintain the low oxygen environment nitrogenase needs. Inside the nodule, the bacteria fix nitrogen. The plant supplies carbon compounds as fuel. Fixed nitrogen moves into the plant. It's a trade in functional terms, though calling it that always makes me want to immediately qualify it. Neither organism is negotiating anything. They're responding to chemistry. The outcome looks like cooperation because it is cooperative in effect, even if there's no intention anywhere in the system. I think that distinction matters. Not for the biology, which works fine either way, but for honesty. From the nodule, fixed nitrogen moves into the plant tissue, into roots, into stems, into leaves. It gets incorporated into amino acids, into chlorophyll, into the enzymes the plant uses to run its own metabolism. When something eats the plant, an insect, a rabbit, eventually maybe something that eats the rabbit, that nitrogen transfers again, gets broken down, reincorporated, moved through another body. When organisms die, decomposers handle the nitrogen in their tissue. Bacteria convert organic nitrogen back into ammonium, a process called ammonification. Other bacteria, the nitrifiers, oxidize ammonium into nitrite and then nitrate, forms that plant roots can absorb directly. And then there are the denitrifiers, bacteria that run the cycle in reverse, converting nitrate back into nitrogen gas under low oxygen conditions, returning it to the atmosphere. The cycle closes. Slowly, imperfectly, but it closes. During the nitrogen assay work in graduate school, I was processing soil samples from several different ecosystems: agricultural fields, remnant prairie, a forest edge. The numbers came back as concentrations, which meant nothing to me until I did the math to figure out what they implied about total nitrogen cycling per hectare per year. And then they meant too much at once, the kind of quantity that briefly makes you feel like your sense of scale is broken. Healthy soil cycles nitrogen through organisms at rates that dwarf what's visible on the surface. The prairie looked quiet. The forest edge looked unremarkable. What was happening in the top 30 centimetres of those soils, in terms of molecular traffic, was not quiet and not unremarkable. It just didn't look like anything. That gap between appearance and activity is one I keep returning to in almost every topic I write about. The most consequential processes are almost always the least visible. The nitrogen in you right now followed a path, something like this, in rough terms, fixed from atmosphere by bacteria at some point, moved into plant tissue, transferred through one or more organisms before reaching your food, broken down in your gut, absorbed, reassembled into your own proteins. How long ago it was fixed is harder to say. Nitrogen cycles on timescales ranging from days to centuries depending on the pathway. Some of it was fixed recently, by bacteria in a field somewhere this season. Some of it has been circulating through organisms for much longer, transferred so many times that the original fixation event is lost to any practical accounting. What's in you is borrowed. The atom doesn't know that. You didn't choose it. But the bacteria that first pulled it from the air and made it available to the rest of the living world, they're still doing that. In soils and root nodules and waterlogged sediments right now, tonight, they're running the same expensive chemistry they've been running for billions of years. And without it, there's no protein, no DNA, no you. That's not dramatic, it's just the situation. Coral looks like rock. That's the first problem. You see it in photographs, or if you've been lucky, in person through a mask, the water pressing gently against your face, and the brain files it under mineral. Texture like stone, rigid, permanent seeming. The colour, when it's there, gets attributed to the structure itself, the way we attribute colour to paint on a wall rather than asking what the paint is made of. The colour is alive, and the structure beneath it is only barely not. A coral polyp is an animal, small, soft-bodied, related to jellyfish in the loose way that reveals something true about how early animal life branched. It secretes calcium carbonate beneath itself, the hard cup it sits in, and that secretion accumulates over time into the skeletal architecture that reefs are built from, the animal on top, the mineral record of its existence below, inside the tissue of the polyp-live algae. Zooxanthellae. The name is unwieldy and I've mispronounced it in ways I won't describe. single-celled photosynthetic organisms that take up residence in coral tissue and stay. This is not incidental. The relationship is so load-bearing for both organisms that separating them, even briefly, tends toward catastrophe. The algae photosynthesize. They use the coral's metabolic waste, carbon dioxide, nitrogen compounds, as raw material. In return, they pass photosynthetic products back to the coral: sugars, glycerol, amino acids. Estimates vary and the specific numbers depend heavily on species and conditions, but somewhere between 70 and 90% of a coral's energy requirements can be met this way, by the organisms living inside its own cells. Think about what that means for a moment. An animal, drawing the majority of its energy not from food it hunts or filters, but from the metabolic output of photosynthetic tenants running quietly inside its tissue. The coral still feeds. It has tentacles, it captures zooplankton, but the algae subsidize it in a way that changes what the coral is, ecologically speaking. It's not quite an animal in the way we usually imagine. It's not quite a plant. It's something the category system wasn't built to handle cleanly. The coral exhales CO2. The algae use it. The algae exhale oxygen. The coral uses that. Back and forth inside a relationship so tightly integrated that the boundary between host and symbiont stops being a useful way to think about either one. Reefs exist because of this. The calcium carbonate secretion that builds reef structure requires energy, and that energy comes largely from the algae. Without the symbiosis, the rate of reef building slows dramatically. The architecture collapses over geological time. The ecosystems depending on that architecture, the fish, the invertebrates, the organisms that shelter and spawn in reef structure, lose their substrate. One relationship running inside individual polyps at the cellular scale, holding up an ecosystem visible from satellites. When water temperature rises beyond a threshold, a degree or two above the typical summer maximum, sustained over weeks, the relationship breaks, the algae leave, or the coral expels them. The mechanics are still debate, which is worth saying. What's clear is that the symbiosis fails under thermal stress, and when it fails, the coral loses its colour. What remains is the white calcium carbonate skeleton beneath translucent tissue. Bleached. The coral isn't dead at that point, but it's running on reserves. If the temperature drops and algae recolonize, recovery is possible. If the stress continues, the coral starves. The skeleton remains. The animal does not. I don't want to linger too long here, because this isn't really that kind of chapter. But it's hard to describe bleaching in purely biological terms and not feel the weight of what that represents at scale. Entire reef systems, thousands of kilometers, bleached in events that have become more frequent, more severe, with less recovery time between them. The biology is the same. The context has changed around it. What I find myself returning to, when I think about coral, is the zooxanthellae specifically. The algae. They don't get the attention the reef gets, or the polyp gets, or the fish do. They're not visible without magnification. Their contribution is chemical, internal, quiet. But the reef is downstream of them, the whole thing. There's a version of the coral story that centers the animal, the skeleton, the ecosystem. That version is true, but incomplete. The more honest version starts smaller, inside a cell, where a photosynthetic organism is running its metabolism and passing the proceeds to its host, and has been doing so for something like 160 million years. The reef is the long-term consequence of that, still running, still exhaling. There is no good way to watch a fungal heifer grow. The extension happens at rates measured in millimeters per hour under good conditions, which sounds fast until you're the one trying to observe it, at which point it becomes the biological equivalent of watching paint dry. Except paint doesn't make decisions. A heifer does in its chemical way. It responds to gradients. It extends toward phosphorus, towards certain root exudates away from toxins and competing fungi. It navigates, slowly, without anything that resembles a nervous system or a center or a self in any sense we'd recognize. I went through a period, maybe 3 years, which my friends found unreasonable, where mycorrhizal networks were essentially all I wanted to talk about. I read everything I could find. I annoyed people at parties. I'm aware this is not a universally relatable experience, but I'm also not apologetic about it, because what I was reading was genuinely changing how I thought about individuality, about where one organism ends and another begins, about whether those categories are as solid as biology textbooks suggest they are. They are not, really. The mycorrhizal relationship is one of the cleaner demonstrations of that. The word mycorrhiza means approximately fungus root. It describes a symbiosis between fungal mycelium and plant root tissue that is, depending on how you count, present in somewhere between 80 and 90% of all land plant species. That number sits strangely in the mind. It suggests not an exception or a specialized adaptation, but something closer to a default state. That the familiar picture of a plant root absorbing nutrients from soil is, in most cases, incomplete. The root is not doing that alone. The fungus extends into the soil beyond the root's reach, far beyond. A single plant can be connected to mycelium that extends meters in every direction, accessing soil volumes the root system could never reach directly. The fungal hyphae, because of their diameter, a fraction of that of a root hair, can penetrate spaces between soil particles that roots cannot enter. They mine the soil in a way roots simply cannot replicate. What they bring back primarily is phosphorus and water. In exchange, the plant supplies carbon, sugars produced through photosynthesis, move down through the stem and out through the roots into the fungal network. The transfer is bidirectional, continuous and adjusted. Research in the last few decades has shown that the plant can to some degree regulate how much carbon it allocates to different fungal partners, apparently favouring partners that deliver more phosphorus. Whether this constitutes something like choice is a question I find interesting precisely because I'm not sure the answer is no. There are 2 broad categories of mycorrhizal association, and they behave quite differently. Ectomycorrhizal fungi, the type associated with many temperate forest trees, including oaks, pines, birches, wrap around the root tip and extend between root cells but don't penetrate into the cells themselves. They form a sheath, a dense fungal layer that the root sits inside, and from this sheath, hyphae extend outward into the soil. Many of the fungi in this group produce the fruiting bodies we recognize as mushrooms. The mushroom, when you see it, is the reproductive structure of an organism whose main body is underground, threaded through the root tips of nearby trees, potentially covering hectares. Arbuscular mycorrhizal fungi, the other major group and the more ancient one, actually penetrate root cell walls and form branched structures inside the cells themselves. These structures, the arbuscules, are where nutrient exchange occurs at close range. They don't produce visible mushrooms. They are obligate symbionts, meaning they cannot complete their life cycle without a plant host. They have never been successfully cultured in isolation. To grow them in a lab, you have to grow them on a plant. They are in some biological sense incomplete without the relationship. This dependency goes both ways in different degrees. Some plants grow poorly in sterile soil without fungal partners. Orchids are perhaps the most extreme case. They cannot germinate without mycorrhizal fungi providing the initial carbon supply to the seedling, because orchid seeds contain almost no nutritional reserves. The seed needs the fungus before it can do anything else. The relationship isn't optional, it's the starting condition. The network aspect is where things become harder to describe cleanly, and I want to be careful here because this particular topic has attracted a lot of language that goes further than the data comfortably supports. What we know: plants in a shared mycorrhizal network can have carbon, nitrogen, and phosphorus transferred between them through fungal connections. This has been demonstrated experimentally with isotopic tracers. The transfer is real. It happens. What's less clear? How much this transfer benefits recipient plants under natural conditions, whether the fungus is an active facilitator or an incidental conduit, and whether terms like communication or cooperation meaningfully describe what's occurring. The popular framing, forests as communities of trees sharing resources through a wood-wide web, is not fabricated, but it's been softened and warmed in ways that occasionally run ahead of the evidence. I say this not to diminish the biology, which needs no embellishment, but because I find imprecision in the direction of the appealing to be its own kind of distortion. The actual situation is strange enough. A fungal network connecting multiple plant root systems, moving carbon from areas of surplus to areas of deficit, without any organism in the system having awareness of any of it, that's already extraordinary. It doesn't need to be more than it is. The timescales involved are worth sitting with. Mycorrhizal associations are ancient. Fossilized evidence places them at approximately 450 million years ago in some of the earliest land plants. This means the relationship predates leaves, it predates seeds, it predates roots as we currently understand them. Early land plants had rhizomes, root-like structures, and the fungi were already there, already involved. The colonization of land by plants may have depended on it. Moving from water to terrestrial environments meant accessing nutrients from a medium, soil or protosoil, that plant tissue alone struggled to exploit efficiently. Fungal partners already adapted to the soil environment potentially made the transition viable in a way it might not otherwise have been. Which means forests, in some sense, are a consequence of this relationship, not just supported by it now, but perhaps initiated by it. The trees we associate with stability and permanence may owe their existence on land to a symbiosis with something most people have never thought about. I keep a field notebook I don't really write in anymore. I carry it out of habit, which I recognize as its own kind of strange attachment. But I remember writing in it during that mycology period. Pages of diagrams, network sketches, notes on papers I was reading, trying to draw what the relationship looked like spatially, trying to get the geometry of it on paper. I couldn't, really. The structure doesn't flatten well. It's three-dimensional and dynamic and differently organized at every scale. What I ended up with was a mess of overlapping lines that looked nothing like the thing I was trying to represent. The hypha I was trying to draw was, at that moment, somewhere in a forest, extending toward a phosphorus source at a few millimeters per hour, responding to gradients, feeding a tree it would never see, not knowing, and this is the part I kept circling in my notes, not knowing anything in any sense of the word. And yet the outcome of that not knowing accumulated across billions of fungal threads and millions of plant-fungal associations was the forest I'd walked through to get to the library. There's no clean conclusion to this. The network doesn't conclude. It extends, retracts, forms new connections, loses old ones, persists through winter in spore form, resumes in spring. The relationship is ongoing in the way that basic chemistry is ongoing, not because anyone is maintaining it, but because the conditions that sustain it keep being met. A hyphae is extending somewhere right now, into soil that's been dark for months, towards something it can sense but cannot see. It has been doing this in one form or another for nearly 500 million years, and underneath the patience of it, if patience is even a word that can stretch that far, something enormous is being quietly held together. Here is something that took me longer than it should have to fully accept. Some bacteria eat rocks, not literally. The word eat is doing dishonest work there, and I know it. But the functional description isn't far off. Certain bacteria extract energy from the oxidation of inorganic compounds. Iron, sulfur, ammonia, manganese. They don't need sunlight. They don't need organic carbon produced by something else. They take electrons from minerals and run them through a respiratory chain. And that process, that dry, almost purely chemical transaction, powers everything they do. Growth, reproduction, the whole biological enterprise. The first time this was explained to me properly, I remember thinking it sounded like something someone had gotten wrong. Iron-oxidizing bacteria are among the more quietly extraordinary organisms on the planet, and they get almost no attention outside of specialist literature, which I find disproportionate every time I think about it. The basic chemistry, ferrous iron, Fe2+, is the reduced form. Iron in this state still has electrons available. Certain bacteria, Acidithiobacillus ferrooxidans is one of the better studied species, strip those electrons from the iron, oxidizing it to ferric iron, Fe3+, the form that precipitates out of solution and turns water that orange-brown colour you see draining from old mines or gathering at the edges of certain bogs, the rusted staining on rocks at the margins of acidic streams. That's biological, or it's partly biological. The bacteria are doing the oxidation, the colour is the evidence. The energy yielded per reaction is small, much smaller than you get from burning glucose or from photosynthesis under good light. To get enough energy to matter, these bacteria have to run the reaction continuously and at scale, which they do, without complaint, which is not a thing bacteria can have, but the word fits anyway. What I find genuinely difficult to hold in mind is how old this metabolism is. Iron oxidation as an energy strategy likely predates the Great Oxidation Event, the same atmospheric shift discussed earlier in this series, when cyanobacteria began flooding the atmosphere with oxygen roughly 2.4 billion years ago. Before that, the oceans held enormous quantities of dissolved ferrous iron in low oxygen conditions. Iron-oxidizing bacteria or their ancestors may have been working that resource for longer than complex life has existed in any form. When oxygen levels rose, the chemistry of the oceans changed. Ferrous iron oxidized abiotically without biological help, and precipitated out in the massive banded iron formations we now mine as ore. But the bacteria didn't disappear. They adapted, shifted to different niches, found low-oxygen microenvironments where the chemistry still worked. Some are still living in conditions that approximate the early Earth, hot, acidic, oxygen-depleted, as if the last 2 billion years happened somewhere else. There's something in that I don't have a clean word for. The sulfur oxidizers are a related story. Bacteria like Beggiatoa, which I remember encountering in a paper during grad school, and then spending an afternoon reading about when I should have been doing something else. Oxidize hydrogen sulfide to elemental sulfur, storing it as granules inside their cells, then oxidizing it further to sulfate when the hydrogen sulfide runs low. They are common in sediments, in hydrothermal areas, in the boundary layers where oxygenated water meets sulfide-rich water below. The sewage treatment facility I visited a few years ago, which I described to people as one of the more genuinely interesting field trips I've taken to varying degrees of belief, had microbial communities doing related chemistry, nitrification, sulfur cycling, organisms running metabolic strategies that predate animal life, deployed in a very contemporary context, doing work that would otherwise require substantial chemical infrastructure to replicate. The engineer giving the tour described the microbial communities as the actual treatment system. The tanks and pipes were just housing. That framing has stayed with me. The rust at the edge of a pond, the orange seep from a hillside cut during road construction, the iridescent sheen on still water near decomposing vegetation. That one is often iron-oxidizing bacteria at the surface, their cellular material forming a thin film that breaks apart when you touch it, unlike an oil slick which reforms. These are biological signatures, evidence of metabolic activity in places we don't usually think of as inhabited. The bacteria producing them have been running the same core chemistry since before the atmosphere was breathable. They don't require the world we've built on top of their work. They were here before it. They are largely indifferent, in the only way things without indifference can be, to what happened next. I want to start with something small, not metaphorically small, actually small, micrometers in diameter, invisible to the unaided eye, drifting in the upper layers of the ocean with no mechanism for directed movement beyond what currents allow. A single cell, no roots, no structure anchoring it anywhere, just a cell with chlorophyll somewhere near the ocean surface where light still penetrates. That cell is producing oxygen right now, tonight. In the dark, it isn't. Photosynthesis requires light, and whatever patch of ocean it's drifting through may be in darkness at this hour. But somewhere on the sunlit side of the planet, cells exactly like it are running photosynthesis continuously, and the cumulative output of those cells, across every ocean, every coastal upwelling zone, every nutrient-rich current, accounts for somewhere between 40 and 50% of all oxygen produced on Earth. I've said that number out loud to people and watched it land wrong. It doesn't feel true. It feels like the kind of statistic that gets exaggerated for effect. But it isn't exaggerated. If anything, earlier estimates ran higher. Phytoplankton is not one thing. That's worth establishing early because the word flattens an enormous amount of biological diversity into a single category defined more by lifestyle than by ancestry. Diatoms, cyanobacteria, again, they qualify. Dinoflagellates, coccolithophores, which build intricate calcium carbonate shells around themselves that sink to the ocean floor when they die and have been accumulating there for so long that the white cliffs of Dover are partly made of them. Green algae, cryptomonads. These organisms are not closely related in the way that, say, mammals are related. Some are prokaryotes. Some are eukaryotes with chloroplasts that were themselves once free-living cyanobacteria, absorbed through a process called endosymbiosis, the same process that gave your cells mitochondria, just applied to photosynthesis instead of respiration. The category phytoplankton is ecological, not phylogenetic. It means photosynthetic, aquatic, microscopic drifting. That's the membership criteria. In graduate school, one of my advisors had a framed satellite image on her office wall, a composite showing chlorophyll concentration in the world's oceans, false coloured so that high productivity showed up in yellows and reds, low productivity in deep blue. I used to look at it while we talked. The productive zones traced the upwelling currents, the continental shelves, the areas where deep, cold, nutrient-rich water rises to the surface and feeds the phytoplankton blooms above. The open ocean, the parts far from coastlines and upwelling zones, ran almost pure blue in the image, oligotrophic, nutrient-poor, sometimes called the ocean's desert, though that framing undersells the biology that does exist there. It just exists at low density and different scale. What struck me, and still does, is the relationship between those patches of colour and the atmosphere above them. The phytoplankton in the yellow zones are pulling carbon dioxide out of the surface water, which drives more CO2 from the atmosphere to dissolve into the ocean to replace it. They are, in aggregate, a carbon pump. Photosynthesizing, dying, sinking. Their bodies carry carbon from the surface to the deep ocean when they sink, sequestering it away from the atmosphere on timescales that range from decades to millennia depending on how deep they sink and how quickly they're remineralized by bacteria on the way down. The biological pump, it's called, which is a functional name but loses something in the translation to engineering language. I've never been on an oceanographic research vessel. This is a gap in my experience I'm aware of, and I mention it because most of what I know about phytoplankton came through other people's data, other people's samples, papers written by researchers who spent weeks at sea collecting water at different depths and running it through equipment I've only seen in photographs. There's a humility required when writing about places you haven't been. The tide pools I can describe from memory, the smell of them, the cold water that got into my boots despite everything, the way the biofilm on the rocks caught the afternoon light, the open ocean I'm reconstructing from literature and satellite images, and the accounts of people who are actually there. What I can say with confidence is that the surface of the ocean is not a passive boundary. It's biologically active in a way that determines atmospheric composition at a planetary scale. The phytoplankton living in the top 100 meters or so of water, the photic zone where light penetrates sufficiently for photosynthesis, a running chemistry that connects the ocean to the sky in both directions. Oxygen out, carbon dioxide in, the exchange is continuous. There is a species worth mentioning by name, partly because the name is satisfying and partly because the biology is startling even by phytoplankton standards. Prochlorococcus, a cyanobacterium, possibly the most numerous photosynthetic organism on Earth. Estimates of its global population run to around 10 to the 27th power, which is a number that stops meaning anything almost immediately, but is worth sitting with anyway. It was not discovered until 1988, which feels recent. It was discovered using flow cytometry, a technique that can detect individual cells in seawater by the way they scatter and absorb light. Before that technology existed, Prochlorococcus was in every tropical and subtropical ocean sample anyone had ever collected, and no one had seen it. It lives at very low light intensities compared to many phytoplankton. Different strains are adapted to different depths, different light regimes. The strains at the surface have adaptations for managing intense light that the deep-dwelling strains lack entirely. The same species, essentially, occupying a gradient from the surface to the lower edge of the photic zone, each strain tuned to its position. A bloom happens when conditions align: nutrient availability, light, temperature, the right absence of grazing pressure, and phytoplankton populations expand rapidly. The ocean changes colour. Satellite images show swirling green and turquoise patterns spreading across hundreds of kilometres of ocean surface. Some blooms are visible from space clearly enough that they were being documented in satellite imagery before researchers fully understood what they were looking at. The oxygen produced during a large bloom is measurable. It diffuses into the atmosphere. It becomes part of what you breathe weeks or months later, after mixing and transport carry it inland. The thought that I find difficult to fully metabolize, and I don't mean this dramatically, I mean it as a genuine description of a cognitive experience, is that the boundary between the ocean and the atmosphere, at the scale of atmospheric chemistry, is partly biological. The ocean is not just a passive reservoir, it is exhaling continuously because of organisms too small to see, running photosynthesis in water that covers 70% of the planet's surface. The sky is partly their work. It has been for a very long time. The smell came back with me. Not a metaphor, I mean, I drove home from that salt marsh field assignment, showered twice, and still caught traces of it the next morning. Hydrogen sulfide is detectable by the human nose at concentrations of less than 1 part per billion. The olfactory system treats it as a priority signal, a warning, something to flag immediately. Which makes sense evolutionarily. High concentrations are toxic. The body doesn't wait to assess. What took me longer to accept is that the smell I kept catching 2 days later, faint in my jacket collar, was evidence of productivity, of a biogeochemical cycle running exactly as it should, of bacteria doing work that the coastal ecosystem genuinely depended on. That reframe didn't come easily. It felt initially like being told something unpleasant was actually fine. But the biology doesn't care how the information lands. Sulfur moves through ecosystems in ways that get far less attention than carbon or nitrogen, and I've never fully understood why. The cycle is ancient. The organisms running it are diverse and metabolically inventive in ways that should attract more interest than they do. The outputs are ecologically significant, and yet sulfur cycling tends to appear in textbooks as a brief chapter near the back after the more celebrated cycles have been given their due. The organisms at the centre of it, sulfate-reducing bacteria, are anaerobes. They live where oxygen is absent, or nearly so. Waterlogged sediments, the deep layers of salt marshes, the bottom of stratified lakes, marine sediments, the digestive tracts of certain animals. In these environments, they use sulfate, SO4 2-, abundant in seawater, as a terminal electron acceptor in their respiratory chain, the same functional role that oxygen plays in your cells. The sulfate gets reduced to hydrogen sulfide. That's the smell. The metabolism is genuinely old. Sulfate reduction may have been operating as an energy strategy before oxygenic photosynthesis existed. These bacteria were running this chemistry in an anoxic world before the atmosphere shifted, and they've continued running it in the anoxic pockets that have persisted ever since. They found the low oxygen corners and stayed there. Desulfur vibrio is probably the most studied genus, which partly reflects how broadly distributed it is. Salt marsh sediments, deep sea floor, oil reservoirs, where sulfate-reducing bacteria are a practical problem for the petroleum industry because hydrogen sulfide is corrosive and their reactivity degrades pipeline infrastructure. The same organism that makes a salt marsh smell the way it does also causes problems in offshore drilling operations. Biology operating across contexts that have nothing to do with each other from a human perspective, and everything to do with each other from a chemical one. The hydrogen sulfide they produce doesn't just accumulate, it gets used. Sulfur-oxidizing bacteria pick up where the reducers leave off. At the interface between anoxic and oxygenated zones, the chemocline in a stratified lake, the surface layer of a sediment, organisms like Beggiatoa, which appeared briefly in the last chapter, and Theobacillus oxidize hydrogen sulfide back to elemental sulfur or sulfate, extracting energy from the reaction. Some of them store elemental sulfur as white granules inside their cells, visible under a microscope, waiting to be oxidized further when hydrogen sulfide runs low. The cycle completes. Sulfate reduced to sulfide in the anoxic layer. Sulfide oxidized back to sulfate at the interface. The same sulfur atom moving between oxidation states, powering different organisms on each leg of the journey. When I try to visualize this in a real sediment column, the way the chemistry changes with depth, millimeter by millimeter, from oxygenated surface water down through the redox gradient into the sulfidic sediment below, what I keep noticing is how precisely different organisms partition that gradient. Each group occupies a zone defined by the chemistry it needs and the chemistry it produces. The whole assemblage is structured by the physics of diffusion and the biology of metabolism, and the result is a layered community operating at a scale you could cover with your thumbnail. The oceanic sulfur cycle has an atmospheric dimension that took me a while to appreciate fully. Certain phytoplankton, particularly a group called coccolithophores, which were mentioned briefly in the last chapter, produce dimethylsulfonopropionate, or DMSP, as an osmolyte, a compound that helps them manage cell volume under changing salinity conditions. When these organisms die or get grazed, DMSP gets released and converted by bacteria to dimethyl sulfide and DMS, which volatilizes and enters the atmosphere. In the atmosphere, DMS oxidizes to sulfate particles that act as cloud condensation nuclei. Water vapor condenses around them. Clouds form. Clouds affect how much solar radiation reaches the ocean surface, which affects phytoplankton productivity, which affects DMS production. Phytoplankton influencing their own light environment through atmospheric chemistry, indirectly without any intention, through a chain of reactions that passes through bacterial metabolism, atmospheric oxidation, cloud physics, and back down to ocean surface irradiance. The cycle proposed around this, that marine phytoplankton regulate climate through DMS production, is called the Cook-Law hypothesis, and its strong form remains contested. But the chemical pathway itself is real and documented. The connection between ocean biology and cloud formation through sulfur chemistry is not speculation. Outside, tonight, if there are clouds, some fraction of the condensation nuclei around which those water droplets formed may have originated as DMS from a marine organism that has no nervous system, no awareness of sky, no relationship to weather in any felt sense. The sulfur left the ocean, oxidized in the atmosphere, seeded a cloud, and the bacteria in the sediment below that patch of ocean are still reducing sulfate in the dark, producing hydrogen sulfide, handing the sulfur back to the organisms waiting at the interface above them. The cycle doesn't pause for night. I want to correct something I believed for an embarrassingly long time. I thought termites ate wood, in the sense that you or I eat food, taking it in, extracting energy from it, using that energy to run a body. The picture in my head was simple. Wood goes in, termite gets the energy, waste comes out. A clean transaction between an insect and a substrate. The reality is considerably stranger, and the correction, when it finally settled in properly, was the kind that makes you reconsider several other things you thought you understood. Termites cannot digest cellulose. Not alone. Not without an elaborate community of microorganisms living in their hindgut that does the actual biochemical work. Some species rely on bacteria, some on protists, some on both. The termite provides the housing, the controlled environment, the mechanical grinding of wood into smaller particles. The microorganisms provide the enzymes. The relationship is so obligatory, so deeply integrated, that when a termite molts, shedding its exoskeleton, which includes the lining of its hindgut, it loses its microbial community and has to reacquire it by consuming the feces or anal secretions of nestmates, which is, depending on your tolerance for biological frankness, either gross or quietly elegant. I've landed on both at different times. The mound itself is the part that I find most difficult to describe accurately, because every framing I reach for turns out to be slightly wrong in a way that matters. It is not a nest in the way a bird's nest is a nest, a structure built for shelter and reproduction and not much else. It is not a building in the sense of a static constructed thing. It is closer to, and I recognize this analogy as imperfect, a body. A structure with internal architecture shaped by function. Gas exchange happens through its walls. The porous outer surface allows CO2 to diffuse out and oxygen to diffuse in, driven by the concentration gradients produced by millions of organisms metabolizing inside. Temperature in the interior is regulated, within a few degrees, across seasonal variation that would be lethal to the colony if experienced directly. The mound breathes, not consciously, not through any mechanism that resembles a lung. But the exchange is real and continuous, and the architecture that enables it was not designed by any individual termite. It emerged from the collective behavior of organisms following chemical signals, depositing material where signals directed them to deposit it, creating a structure no single individual could have planned or understood. The fungus-growing termites, primarily the subfamily Macrotermitinae, found across sub-Saharan Africa and parts of Asia, add another layer that shifts the whole picture again. These species don't rely solely on gut microorganisms to process wood. They cultivate fungus. Inside the mound in dedicated chambers, workers deposit chewed plant material, wood, grass, leaf litter, onto which they inoculate spores of Termitomyces, a fungal genus found nowhere outside termite mounds. The fungus grows on the substrate, breaking down lignin and cellulose, using its own enzymatic toolkit. The termites then eat the fungus and the partially degraded plant material the fungus has been growing on, and pass it through their gut where microbial communities continue the breakdown. It is a two-stage external digestive system. The mound contains a gut before the gut. Termitomyces produces mushrooms, some of the largest mushrooms in the world, emerging from the mound after rain. The mushrooms disperse spores. New colonies acquire the fungus when a founding queen carries spores with her. The association is ancient, somewhere around 30 million years, and the fungus has co-evolved with its termite cultivators to the point where the relationship is obligate for both. Termitomyces cannot survive without the mound conditions the termites maintain. The fungus-growing termites cannot thrive without the fungal processing. Two organisms, neither complete without the other, building a structure that houses them both. I spent an afternoon once. This was during a visit to a natural history collection, not fieldwork, which I should admit, looking at cross-sections of termite mound material under magnification. The internal architecture was more intricate than I'd expected from photographs. Channels of varying diameter, chambers with walls of different thickness and porosity. The material itself was a composite. Soil particles, termite secretions, organic matter, fungal mycelium in the fungus-growing species. It had structural properties that engineering materials researchers have actually studied because the mound maintains its integrity under rain, heat, and significant external pressure. What I kept thinking looking at it was how much information was encoded in the structure, not consciously stored, not planned, but present, in the channel diameters that regulated gas flow, in the chamber placement that maintained temperature, in the wall porosity that balanced moisture retention against ventilation. Every functional property of the mound was an accumulated consequence of individual behavioral responses to chemical signals across generations of workers, none of whom had any representation of the whole. The mound knows things in a functional sense that no termite knows. The ecological contribution of termite mounds extends well beyond the mound boundary. In African savannas, termite mounds are islands of elevated nutrient concentration in nutrient-poor landscapes. The foraging activity of the colony brings organic material from the surrounding area back to the mound. Microbial processing releases nutrients into the mound soil. The mound soil is richer in nitrogen, phosphorus, and carbon than the surrounding matrix. Plants growing on or near mounds show different growth patterns. Grazing animals concentrate around mounds during dry seasons because the vegetation is better. A single mound influences the ecology of an area far larger than its footprint. A landscape with many mounds, a termite savanna, which covers significant portions of sub-Saharan Africa, is a landscape structured by the collective foraging and building activity of these colonies, operating at scales no individual colony is aware of. The mound processes wood, the mound processes carbon, the mound processes the landscape, the gut microbiome of a termite hindgut, and I recognise this is a lot to sit with, so I'll keep it brief, contains bacterial diversities that rival some of the most complex microbial communities known. Hydrogen-producing fermenters, hydrogen-consuming acetogens and methanogens, spirochetes performing unusual metabolic functions. The methanogens are worth noting specifically. Termites produce methane as a byproduct of hindgut fermentation, and global termite methane emissions are a real, if contested, contribution to atmospheric methane budgets. An insect, a gut, a mound, a fungus, a landscape, an atmosphere. The connections don't stop at the mound wall, they rarely do. Tonight, in a savanna somewhere, a mound is warm from the inside, not from the sun. The temperature is maintained by the metabolic heat of millions of organisms, and it will stay within a few degrees of optimal through the night while the air outside drops. Workers are moving through channels in the dark, guided by pheromone trails they cannot see, doing tasks they cannot conceptualize, maintaining a structure none of them understand. The structure understands itself in the only way structures can, through function, through persistence, through the quiet, continuous fact of still working. There's a particular kind of cognitive vertigo that comes from trying to think in geological time. Not astronomical time, I find that easier somehow, because the distances are so abstract they stop feeling like time at all and become more like a spatial problem. Geological time is worse. It's close enough to feel like it should be comprehensible, but long enough that the numbers stop meaning anything concrete about a third of the way through the attempt. 300 million years. 360 million. The Carboniferous period, named for the carbon it left behind, running from roughly 358 to 298 million years ago, during which something happened that altered the planet's carbon chemistry for the rest of geological history. I want to approach this carefully because the temptation with deep time narratives is to compress them into drama, to make the Carboniferous feel like an event, a turning point with a shape and a resolution. It wasn't. It was 60 million years of biology operating under conditions that no longer exist, producing consequences that accumulated so slowly no organism living through them could have detected the change. That's the thing about geological time that I keep failing to adequately represent in my own thinking. Nothing felt like anything. There was no threshold moment, just continuous biological activity, sustained across spans that dwarf the entire existence of our genus by factors that are difficult to say out loud without feeling slightly ridiculous. The Carboniferous world was wet, warm, heavily forested, not with the trees we know, but with lycopsids, giant club mosses that grew to 30 or 40 meters, with tree ferns, with early seed plants moving through their own evolutionary experiments. The forests were dense, and the organic productivity was high, and organisms were dying in enormous quantities and falling into the swampy, waterlogged environments that covered much of the land surface. And here is the part that I find genuinely difficult to move past without pausing. Lignin, the structural polymer in wood that makes it rigid and resistant to decay, was relatively new. Plants had only recently evolved the capacity to produce it in quantity. The organisms capable of breaking it down efficiently, the white rot fungi and bacteria we discussed in the Decomposition chapter, had not yet fully caught up. The evolutionary arms race between lignin production and lignin degradation was, in the Carboniferous, running heavily in favour of the plants. Dead plant material accumulated, faster than it decomposed. In the waterlogged, low-oxygen conditions of Carboniferous swamps, decomposition slowed further. The anaerobic environment limited the activity of even the decomposers that were present. Organic material compressed under the weight of subsequent accumulation. Water was squeezed out. Carbon was concentrated. Over millions of years, under heat and pressure, that compressed organic material became coal. I want to sit with what coal actually is for a moment, because I think the familiarity of the word has made it harder to think about clearly. Coal is biological material. It is the compressed, chemically altered remains of organisms, mostly plants, partly the microbial communities that were partially decomposing them, that lived during the Carboniferous and Permian periods. When you burn coal, you are oxidizing carbon that was fixed from the atmosphere by photosynthesis somewhere between 300 and 360 million years ago, and that has been sitting in reduced form underground ever since. The carbon dioxide released by that combustion was last in the atmosphere before the first dinosaurs existed, before flowers existed, before birds existed, before grass existed. It was fixed into organic molecules by organisms doing what organisms do, running their metabolisms, building their tissues, living their lives, and it stayed fixed, stayed underground, stayed out of the active carbon cycle for a span of time so long that the continents were in different positions when it began. The Atlantic Ocean didn't exist yet, not even close. The land masses that would become Europe and North America were pressed together, and the swamp forests in what would eventually become the Appalachian region and the coal fields of Wales and Belgium were part of the same continuous ecosystem, separated now by an ocean that opened up 100 million years after the coal was already forming. There's a question I find myself circling which I'll admit I've never fully resolved to my own satisfaction. How much did the carbon burial of the Carboniferous change the atmosphere in real time? The removal of CO2 from the atmosphere through organic burial should have had a cooling effect, and there is evidence for glaciation at the end of the Carboniferous, the Permo-Carboniferous glaciation, one of the major ice ages in Earth's history. The correlation between carbon burial and cooling is real enough that it appears in multiple lines of evidence, but the causality is genuinely complex. Atmospheric oxygen also rose during the Carboniferous, possibly to levels significantly higher than today. Some estimates suggest 35% compared to the current 21%, driven by the same organic burial that was removing carbon. High oxygen levels had their own effects on fire regimes, on animal physiology, on what body sizes were viable for different groups of organisms. I've read 3 competing models for Carboniferous oxygen levels, and I'm still not entirely sure which one I find most convincing. This might be worth checking before the final script. The system was coupled in ways that make clean causal statements difficult, which is honestly the normal situation in Earth system science, and I've made peace with it mostly. The honest answer to many questions about deep time is, something like this happened, for reasons something like these, with consequences something like those, and the uncertainty gets larger as you zoom in. The carbon stayed underground for 300 million years. It did nothing. It participated in nothing. It was not part of any food web, not cycling through any organism, not influencing any atmospheric chemistry worth measuring. It was just there, compressed, reduced, inert in the geological sense. And then, in a span of time that is, relative to how long it was underground, essentially instantaneous, 250 years, roughly, of industrial extraction and combustion, started moving again. The same carbon that was removed from the active cycle slowly, over tens of millions of years, is being returned to it at a rate with no geological precedent in the timeframes we're talking about. I don't say this to be pointed. I say it because the biology of it is real and worth understanding on its own terms, separate from any argument about what follows from it. The carbon cycle has a deep history. The coal seams currently being mined represent a specific chapter in that history, a period when decomposition failed to keep pace with production, when organic material accumulated instead of cycling, when carbon left the active biosphere for geological storage. That chapter took 60 million years to write. There are organisms alive today that are doing, in small ways, what the Carboniferous forests did at scale. Peatlands, bogs, fens, the vast wetlands of the boreal zone across Siberia and Canada accumulate organic material in waterlogged, low-oxygen conditions that slow decomposition. The carbon accumulates slowly. A metre of peat might represent thousands of years of plant growth and partial decomposition. Peatlands cover around 3% of the Earth's land surface and hold somewhere between 500 and 600 gigatons of carbon, more than all the world's forests combined, stored in a substrate that looks from the surface like wet ground. The same basic dynamic, organic productivity outpacing decomposition, carbon accumulating in reduced form, a slow, imperfect biological carbon capture running on no energy source other than photosynthesis and the chemistry of waterlogged soil. No one designed it. No organism is trying to store carbon. The sphagnum moss that dominates many peat bogs produces compounds that inhibit microbial decomposition of its own tissue, an adaptation that likely evolved for other reasons, and that incidentally makes it extraordinarily good at accumulating in layers that compress into peat over millennia. Evolution producing, as a side effect, a carbon sequestration system of planetary significance. I think about the Carboniferous forests sometimes when I'm outside in a way that's hard to describe without sounding more meditative than I actually am. Not romanticizing it, the Carboniferous wasn't a paradise. It was a functioning ecosystem with its own pressures and extinctions and weirdness. But thinking about what the ground is made of, what's underneath. The carbon that's still down there in seams and deposits, the compressed remains of organisms that photosynthesize sunlight that fell on a planet arranged entirely differently than the one I'm standing on. It's a lot to hold. Usually I can't hold it for long before it slips back into abstraction. But for a moment, sometimes it's there. The depth of it. The enormous patience of carbon going nowhere. Waiting underground in the dark for something to come along and move it. I'll be upfront about something. I came to bioluminescence late and slightly reluctantly. It felt for a long time like the crowd-pleasing corner of marine biology. The thing you put in a documentary when you need something visually striking, glowing plankton, glowing jellyfish. The anglerfish with its dangling light lure, which I admit is genuinely interesting but had been described to me so many times by the time I encountered it in any depth that I'd developed a mild aversion to it the way you develop an aversion to a song you've heard too many times in the wrong contexts. What changed my mind was a paper on the biochemistry of the luciferin-luciferase reaction in unrelated lineages, and the realization that bioluminescence had evolved independently somewhere between 40 and 70 times across the tree of life, possibly more. The number kept getting revised upward as molecular phylogenetics improved. 40 to 70 independent evolutionary origins of the same basic phenomenon. Light produced by a living organism through a chemical reaction. That stopped being crowd-pleasing and started being strange in a way I found more interesting. The chemistry, stripped to its essentials, involves a substrate molecule, luciferin, though different lineages use chemically distinct luciferins, being oxidized in a reaction catalyzed by an enzyme called a luciferase. The oxidation releases energy, and some fraction of that energy is released as a photon of visible light rather than as heat. The reaction requires oxygen. It produces, depending on the organism, light ranging from blue-green to yellow, with blue-green being dominant in marine organisms. And that's not coincidental, because blue-green wavelengths penetrate seawater more effectively than red or yellow, which are absorbed quickly at depth. The efficiency is striking. A chemist I met briefly at a conference described it in terms I'm probably slightly misremembering as one of the most efficient light-producing reactions known, somewhere above 90% of the energy released going to light rather than heat. A comparison to an incandescent bulb which loses most of its energy as heat was made. I've since looked this up and the numbers vary significantly depending on the species and conditions, but the basic point holds. Biological light production is, in certain organisms, extraordinarily efficient in ways that materials scientists have studied specifically to understand. The independent evolution problem is the part that I keep returning to. When a feature evolves once, you can tell a fairly clean story about it. A mutation occurred. It conferred some advantage. It spread. The story might be complicated in the details, but the basic shape is tractable. When a feature evolves 40 or 70 times independently, in organisms with no common ancestor that had the feature, something different is being said. The chemical pathway to bioluminescence is apparently accessible from many different evolutionary starting points. The raw materials, oxidizable organic compounds, oxygen, proteins that can catalyze the reaction are widely available in living systems. The step from there to bioluminescence is, apparently, not as large as the rarity of the phenomenon might suggest. What this implies, and I find this genuinely interesting as a question about evolution, is that bioluminescence may represent a kind of low-hanging evolutionary fruit that gets repeatedly discovered rather than a difficult innovation that required special circumstances. The ocean is full of organisms that haven't evolved it, but many of the ones that have seemed to have arrived there through different routes, using different molecules in different geological periods. Some of those origins may have been accidents, not in a dismissive sense. Accidents in evolution can be the most consequential things that happen, but there are bioluminescent organisms where the light appears to be a byproduct of oxidative chemistry. The organism was running for other metabolic purposes entirely. The light wasn't the point. The oxidation was the point. The light just happened, and then, in some lineages, it became the point. The deep ocean at night, which is to say the deep ocean always, because light from the surface doesn't reach below about 1,000 meters under the best conditions, and most of the ocean is deeper than that, is not dark in the way a closed room is dark. Estimates of what percentage of deep-sea organisms produce bioluminescence vary, but the numbers are high. 76% of marine organisms in one depth range studied off the California coast. Other surveys have returned similar figures for different regions. In the mesopelagic zone, the twilight zone between 200 and 1,000 meters, bioluminescence is pervasive enough that the darkness is intermittently interrupted by the flash of a dinoflagellate disturbed by movement, by the steady glow of a bacterial colony on a sinking piece of organic matter, by the counter-illumination of fish producing light on their ventral surface to match the dim light coming from above and eliminate their shadow from predators below. That last one, counter-illumination. An organism producing light specifically calibrated in intensity and direction to make itself harder to see. The light exists to create an absence of visible contrast. It is camouflage made from photons. I find that quietly extraordinary in the specific way I reserve for things that took me a moment to fully pass. Not immediately impressive. Impressive after the mechanism settles in. Dinoflagellates deserve a specific mention because they are likely the bioluminescence most people have encountered, or could encounter, without knowing what they were seeing. The blue flash in a wave at night. The glow in the water disturbed by a boat's hull. The light that appears when you move your hand through water in certain coastal conditions. That's dinoflagellates, single-celled organisms, many of them photosynthetic during the day, that produce bioluminescence when mechanically disturbed. The light is produced in organelles called scintillons, and the trigger is a rapid drop in pH caused by the mechanical disturbance opening ion channels. Why they do this is still somewhat debated. The burglar alarm hypothesis suggests the flash attracts larger predators that eat whatever was disturbing the dinoflagellate, using bioluminescence to summon a defender. There's experimental support for this. There are other hypotheses. The honest answer is that function is harder to establish than mechanism, and for many bioluminescent systems the evolutionary function remains partially open. The luciferin in fireflies is different from the luciferin in marine organisms. Different chemical structure, different evolutionary origin, similar enough outcome that we use the same word for both. 40 to 70 times something found its way to light. Most of those paths led somewhere, to communication, to predation, to camouflage, to luring prey into darkness. Some of them may have led nowhere in particular, just oxidation, happening in the deep water, releasing photons into a place where almost nothing could see them. Light made briefly and then gone, in an ocean that wasn't watching. I've put my hands into a lot of soil over the years. Not delicately, I mean actually pushed into it, during that field semester cataloguing soil invertebrates, crouching in humid forest with a trowel and a collection jar, pulling apart the upper layers of the O horizon and the A horizon, looking for springtails and mites and millipedes, and occasionally in the wetter sections near a stream edge, earthworms. The soil in that forest had a smell I still recognize when I encounter it. Dense, faintly see it, alive in a way that dry or compacted soil is not. It smelled like something was happening in it, which of course something was continuously, in every cubic centimetre. The earthworms, when I found them, felt significant in a way I couldn't immediately justify scientifically. They were large, some of them, substantial, moving through the soil with a muscular, deliberate quality that seemed out of place given that they have no skeleton, no rigid structure, nothing to push against except the soil itself and the hydrostatic pressure of their own fluid-filled body. I put them in the collection jar and felt faintly like I was handling something important. An earthworm's digestive system is simpler than yours in terms of structural complexity and more consequential than yours in terms of ecological effect, which is the kind of statement that requires some unpacking. The basic anatomy: a tube running from mouth to posterior opening. Soil enters at one end. The earthworm moves through soil by ingesting it, though this is a simplification of a more varied feeding behaviour, passes through a muscular gizzard that grinds it mechanically, moves through the intestine where chemical and microbial digestion occurs, and exits as casts. The casts, and I will use the polite word throughout, having used less polite ones internally, are chemically and biologically distinct from the soil that entered. The pH shifts. In acidic soils, earthworm casts tend to be less acidic than the surrounding matrix. Microbial biomass in casts is higher than in unprocessed soil, sometimes by an order of magnitude. Nitrogen, phosphorus, and potassium in plant-available forms are elevated. Soil aggregation, the tendency of soil particles to clump into stable structures that hold moisture and resist erosion, is enhanced by the mucus and organic compounds the earthworm secretes during passage. What comes out is not waste in the sense we usually mean. It's processed material, improved in most respects for the purposes of plant growth. There's a quality to this that I think gets undersold in the standard account of earthworm ecology, which tends to lead with Darwin. He spent 40 years studying earthworms, published a book on them in 1881. It was reportedly his favourite work, and then moved fairly quickly to the headline finding that earthworms are good for soil, which they are, but the mechanism is where the interest lives. The earthworm gut is not sterile. It contains microbial communities that differ from those in the surrounding soil, selected for by the gut environment, the pH, the oxygen levels, the available substrates. As soil passes through, these microbial communities act on organic material in ways the earthworm's own enzymes cannot. The earthworm provides the transit conditions, the microorganisms provide much of the chemistry. What the earthworm extracts nutritionally comes partly from microbial biomass, partly from the organic material the microbes have partially broken down. The earthworm is running an external digestion strategy with an internal location. The soil is the substrate. The gut microbiome is the primary processing system. The worm itself is providing the infrastructure and collecting a fraction of the output. I find this framing more accurate than the simpler version, and also more interesting, which in my experience is usually a sign that the framing is closer to what's actually happening. The physical effects are as significant as the chemical ones. Earthworm burrows, channels ranging from a few millimeters to over a centimeter in diameter, running vertically and horizontally through the soil profile, alter the physical structure of the soil in ways that affect water infiltration, gas exchange, and root penetration. A soil with high earthworm activity has higher porosity. Water moves through it more readily. Roots find less resistance. During heavy rain, soils with intact earthworm burrow networks drain faster, which reduces runoff and erosion. The burrows are also habitats. Microbial activity is elevated along burrow walls, the drillosphere, which is the technical term for the zone of soil directly influenced by earthworm activity, including the burrow walls, the casts, and the mucus trails. Different microbial communities colonize this zone than the bulk soil beyond it. The earthworm creates ecological real estate as it moves. The compost heap I turned twice a week during that university semester was full of them in the lower layers, where the temperature had dropped from the hot centre of microbial activity to something more moderate. I'd lift a section of partially processed material and find earthworms already working through it. Not many, but present, moving through what had recently been kitchen scraps and dry leaves in a state of active transformation. The heap was warm from microbial metabolism. The earthworms were working the zones where the temperature allowed it. The output was going somewhere, into the lower layers, into the soil beneath the heap. And the gardeners using the compost were benefiting from biological processing that had already happened before they applied anything. No one had designed this sequence. It was just what happened when organic material, moisture, and biological communities were put in proximity. Globally, earthworm activity moves an amount of soil that is difficult to visualize without feeling briefly absurd. Darwin estimated for a single English field something in the range of 10 tons of soil passing through earthworms per acre per year. More recent estimates vary widely by ecosystem and earthworm density, but the basic scale has held up. 10 tons per acre per year, moved through digestive systems, chemically transformed, deposited at the surface or redistributed through the profile. The field looks still from above. Nothing about the surface suggests this is happening, but push your hand into the soil, into the right kind of soil, and you'll feel the texture of it, loose, crumbly, structured in a way that compacted or degraded soil is not, and that texture is partly biological, partly the accumulated effect of bodies moving through it, digesting it, restructuring it, for longer than our species has existed. The soil that grows things has, in many cases, already been something's gut. There's a photograph I've seen reproduced in enough oceanography papers that I've stopped being able to remember where I first encountered it. A satellite image of the Southern Ocean, false-coloured for chlorophyll concentration. Most of the frame runs a deep, uninterrupted blue. the colour of water that has almost nothing photosynthetically active in it. And then, in one section, a bloom. Green spreading through the blue in shapes that follow current boundaries, irregular, alive-looking in a way that satellite images don't usually manage. The contrast is stark enough that you don't need a scale bar to understand something significant is happening in one part of the frame and not the other. What separates the green from the blue in many parts of the Southern Ocean is not temperature, not light availability, not the presence or absence of the organisms that could bloom if conditions allowed. What separates them often is iron, specifically the near total absence of dissolved iron in the surface waters of the open Southern Ocean and its presence in trace quantities wherever the bloom is occurring. Trace quantities, nanomolar concentrations, amounts so small that measuring them accurately requires extraordinary precautions against contamination. A researcher's skin, a steel instrument briefly dipping into a sample, the hull of a ship. All of these introduce enough iron to confound the measurement. The ocean is not iron poor in the sense of having none. It is iron poor in the sense that the amounts present are almost undetectable by the standards of most analytical chemistry, and those almost undetectable amounts are the difference between biological productivity and biological silence. Iron is the fourth most abundant element in the Earth's crust. This fact and the previous paragraph sit in uncomfortable proximity, and the discomfort is justified. There's a genuine puzzle here that took oceanographers a while to fully accept. The ocean is iron limited in many regions because iron is not very soluble in oxygenated seawater at typical ocean pH. It oxidizes readily to ferric iron, which precipitates out of solution. The forms that remain dissolved are present in vanishingly small concentrations. In the open ocean, far from continental shelves where rivers and sediment supply iron, and far from atmospheric dust inputs that deposit iron-containing particles from arid continental regions, surface waters can be genuinely depleted. The iron that does arrive gets stripped out by phytoplankton within the photic zone, and what sinks with their bodies when they die doesn't easily return to the surface without the physical mixing that upwelling currents provide. The hypothesis that iron availability limits primary productivity in large ocean regions, the iron hypothesis, associated primarily with the oceanographer John Martin, who in 1990 reportedly said, with a degree of provocation, that given enough iron he could give you an ice age, was genuinely controversial when first proposed. It required accepting that a micronutrient, present in concentrations measured in billionths of a gram per litre, was controlling productivity across millions of square kilometres of ocean. The iron fertilisation experiments that followed, deliberately adding dissolved iron to patches of open ocean and watching what happened, confirmed the basic relationship. Phytoplankton bloomed. Chlorophyll concentrations rose. The blue turned green measurably in response to iron additions that were themselves tiny in absolute terms. The connection to your blood is not metaphorical, and I want to be precise about it because imprecision here loses what makes it interesting. Iron in your blood is bound to hemoglobin, specifically to the heme group, an organic ring structure with an iron atom at its centre. That iron atom is what binds oxygen. When you breathe, the iron in your red blood cells picks up oxygen in the lungs and releases it in the tissues. The chemistry depends on the iron being in the correct oxidation state, ferrous Fe2+, and on the protein environment of the heme group that keeps it in that state and controls how tightly it holds oxygen. Iron in phytoplankton is doing related work. It's present in the electron transport chains of photosynthesis and respiration, in the iron-sulfur clusters and cytochromes that move electrons through the reactions that ultimately produce ATP. Photosystem 1, the reaction center in oxygenic photosynthesis, contains multiple iron atoms. Nitrogenase, the nitrogen-fixing enzyme discussed in the nitrogen chapter, is iron-dependent. Catalase, which breaks down hydrogen peroxide and is present in virtually all organisms that deal with oxygen, requires iron. Iron is woven into the biochemistry of aerobic life in ways that reflect its central role in electron transfer chemistry. The same properties that make it useful in biological systems, its ability to cycle between oxidation states to donate and accept electrons, also make it problematic in terms of ocean solubility. Biology uses iron because iron chemistry is versatile. The ocean loses iron from surface waters because iron chemistry is reactive. I ran into a paper during grad school. I was looking for something else, as is usually the case with papers that end up mattering, on dust transport from the Sahara and its relationship to Amazon productivity. The basic finding: iron-containing dust from the Saharan desert is transported across the Atlantic Ocean by wind, deposits into the Amazon basin in quantities that supply a significant fraction of the phosphorus and iron inputs to the rainforest ecosystem. The Sahara feeding the Amazon, not through any biological connection, not through any shared water system, through the atmosphere, through dust. I had to read it twice, not because the mechanism was unclear, but because the scale of it, continent to continent, desert to rainforest, carried by wind across thousands of kilometers of ocean, felt like the kind of thing that should have been more prominently mentioned in every ecology course I'd taken. It wasn't. I filed it in the category of things that should make everyone feel better about how interesting the world is, and then largely failed to bring it up in social situations because the entry point is awkward. The iron cycle in the ocean interacts with the carbon cycle through productivity in ways that have attracted serious attention in the context of past climate transitions. During glacial periods, ice core records show higher dust deposition in many ocean regions. More dust means more iron input to surface waters. More iron means more phytoplankton productivity. More phytoplankton means more carbon drawn down from the atmosphere and exported to the deep ocean when the phytoplankton die and sink, the biological pump running harder. More carbon in the deep ocean means less in the atmosphere. Less atmospheric CO2 means less greenhouse warming, which feeds back into the glacial conditions that produce the dust in the first place. Glacial cycles and iron availability and ocean productivity and atmospheric carbon, all connected through a chain of mechanisms that cross the boundary between biology, chemistry, and physics so frequently that treating them as separate disciplines becomes an obstacle to understanding rather than an aid. The dust is geological, the response is biological, the consequence is atmospheric. The same iron atom moves through all three. Somewhere tonight, a phytoplankton cell is iron limited in the open ocean. It has the light, it has the nutrients, mostly nitrogen, phosphorus, in workable concentrations. What it doesn't have is enough iron to run its electron transport chains at full capacity. Its growth rate is suppressed. The bloom that might have been isn't. And somewhere else, closer to a continental shelf where a river is depositing sediment, or downwind of a dust source, a bloom is happening. The water is green, oxygen is being produced, carbon is being fixed into organic molecules that will either be eaten or will sink. The same ocean, radically different biological activity, separated by a nutrient present in concentrations smaller than anything we encounter in daily life. Your blood is red because of iron, absorbing certain wavelengths of light within the heme group. Oxygenated hemoglobin absorbs blue and green and reflects red. Deoxygenated hemoglobin absorbs slightly differently, which is why venous blood is a darker red rather than blue. A misconception so persistent that I've given up correcting it in casual conversation, though I'll correct it here. Productive ocean water is green because of chlorophyll, which absorbs red and blue wavelengths and reflects green. The iron in the chloroplast makes the electron transport work. The electron transport makes the photosynthesis work. The photosynthesis makes the chlorophyll worth having. Iron in your hemoglobin. Iron in the phytoplankton's photosystem. The same element, doing recognizably related chemistry, in a cell in your blood and a cell in the upper ocean. The colour of your blood and the colour of a productive ocean are both, in their different ways, signatures of iron doing what iron does in the presence of oxygen and the demands of living chemistry. Neither of them knows the other exists. I want to make a small confession. For a long time I thought of fermentation as a human technology. Not consciously, I never would have said it out loud, but the mental category I kept it in was the same one I kept bread and cheese and wine in. Things people made, processes with histories you could trace through civilizations, through monasteries and bakeries and the particular corners of particular kitchens where sourdough starters were kept alive across generations. It took longer than it should have to fully dislodge this. The organisms doing the fermentation predate human kitchens by something in the range of 3 billion years. The process was not discovered or developed or refined by us. We found it already running and inserted ourselves into it, which is a different thing entirely, and the distinction matters more than it might initially seem. Fermentation in the biochemical sense is not defined by the presence of alcohol or the production of anything that smells interesting. It's defined by what's absent. Oxygen, or more precisely the use of an external electron acceptor. In aerobic respiration, the kind your cells run most of the time, the kind that requires the oxygen you inhale, electrons removed from glucose during glycolysis are ultimately passed to oxygen at the end of the electron transport chain. Oxygen is the final acceptor. It gets reduced to water. When oxygen isn't available, that pathway closes. The electron transport chain stalls because there is nothing at the end to accept the electrons. But glycolysis, the first stage of glucose breakdown, occurring in the cytoplasm before the mitochondria get involved, can still run, provided the cell has a way to regenerate the electron carrier molecule NAD+, that glycolysis requires to keep operating. Fermentation is that regeneration system. The cell takes the pyruvate produced by glycolysis and converts it to something else, lactate, ethanol, various organic acids depending on the organism, in a reaction that incidentally regenerates NAD+, and allows glycolysis to continue. The organism gets a small inefficient trickle of ATP. The byproduct, the ethanol, the lactate, the acid, gets excreted as waste. The alcohol is waste. I find this clarifying in a way that took me a beat to appreciate the first time someone explained it to me cleanly. Yeast is not making wine. It's dumping a metabolic byproduct into its environment while trying to survive an oxygen-poor situation. And we have organized enormous industries around collecting that byproduct. Yeast, and I'm talking primarily about Saccharomyces cerevisiae, the species most associated with bread and wine and beer, though the category yeast covers enormous diversity, is a facultative anaerobe. It can run aerobic respiration when oxygen is available, and it does efficiently. When oxygen runs out, it switches to fermentation. The switch is regulated, and the regulatory details involve some elegant biochemistry, but the ecological logic is simple. The organism encounters a situation where its preferred metabolic pathway is unavailable, and it runs the backup instead. The backup produces ethanol, which is toxic. At high enough concentrations, ethanol kills the yeast producing it. Some wine fermentations slow and stall not because the sugar runs out, but because the yeast has poisoned its own environment. The upper tolerance varies by strain. Wine yeasts have been selected, over centuries of human cultivation, for higher ethanol tolerance than wild strains. But the basic dynamic is unchanged. The organism is producing something that will eventually kill it as a metabolic necessity because the alternative is not producing ATP at all. There's something in this that I've never found a satisfying way to describe without reaching for language that anthropomorphizes in ways I'm not comfortable with. The yeast is not in distress. It is not making a trade-off in any conscious sense. It is running the chemistry available to it under the conditions present. The tragedy is a narrative we impose. The biology is just the biology. The timeline matters here, and it's worth being slow about it. The earliest evidence for fermentation as a metabolic strategy pushes back to organisms that existed before the Great Oxidation event, before the atmosphere had significant oxygen. For those early organisms, fermentation wasn't a backup system engaged when oxygen ran out. Oxygen wasn't there to begin with. Fermentation was the system. Glycolysis and some version of fermentation to regenerate NAD+ was the metabolic core of early life, running in an anoxic world, extracting what energy was available from organic compounds. The electron transport chain, with its far greater efficiency, came later, enabled partly by the rising oxygen that cyanobacteria were producing. The organisms that developed aerobic respiration gained an enormous energetic advantage, but fermentation didn't disappear. It persisted as a fallback, encoded in the biochemistry of organisms that also evolved aerobic pathways. It's still there in your muscle cells, running when oxygen delivery can't keep up with demand. The burning sensation in muscles pushed past their aerobic capacity is partly the effect of lactate accumulation. Your cells running their ancient backup chemistry. The same pathway. In you, in yeast, in bacteria that haven't changed significantly in billions of years, running the same core reactions in the same sequence, because those reactions were present in something close to a common ancestor of all life currently on this planet. I've made bread by hand a few times, which I mention not because it's a particularly distinguished thing to do, but because the experience of it is biologically instructive in a way that reading about it is not quite the same as. The moment when a dough that has been sitting covered on a counter starts to smell different, yeasty, faintly alcoholic, alive, is the moment you're detecting fermentation byproducts diffusing through flour and water. The CO2 being produced by the same metabolic pathway is inflating small bubbles in the gluten network, making the dough rise. The ethanol mostly volatilizes in the oven. The organic acids produced by bacterial fermenters also present in sourdough cultures, mainly lactobacillus species, stay and contribute to flavor. You're eating the metabolic waste of organisms that were active for a few hours in your kitchen. And those organisms were running chemistry that predates the kitchen, the building, the city, the species, the genus. Tonight, somewhere in a compost heap, in a waterlogged sediment, in the gut of an animal in the dark, fermentation is running. In the absence of oxygen, or despite its presence, cells are regenerating NAD, plus the only way available, dumping byproducts, extracting a small yield of energy from glucose. It's not elegant, exactly. It's ancient and inefficient, and it produces things the cell immediately discards. But it works. It has always worked. And everything that runs the more sophisticated chemistry learned it from something that only had this. The assumption felt so obvious it didn't seem like an assumption. All life depends on sunlight. Photosynthesis at the base, everything else built on top of that foundation. Plants capture solar energy. Herbivores eat the plants. Carnivores eat the herbivores. Decomposers process what's left. The sun is the engine. Everything downstream is just the engine's consequence. I learned this early, in the way you learn things that don't feel like they will need to be verified, because they're simply the shape of how the world works, foundational, unexamined, almost invisible as an assumption because it's never been usefully wrong before. In 1977, a research submersible called Alvin descended to the Galapagos Rift, a section of the mid-ocean ridge system in the eastern Pacific, at a depth of around 2,500 meters. No sunlight reaches 2,500 meters. The water at that depth is cold, dark, under pressures that would be lethal to any unprotected air-breathing organism. The researchers aboard expected, if they found anything at all, the sparse, slow communities typical of the deep-sea floor. Organisms adapted to scavenging whatever organic material sinks from the productive surface waters above. What they found instead was a vent field generating warm water, and around it clustered in densities that shouldn't have been possible in the deep sea, an ecosystem. Tube worms over 2 meters long, clams, mussels, crabs, shrimp, fish, an entire food web thriving in complete darkness, at depth, in the absence of photosynthetic primary production. Nobody had predicted it. The assumption had been so total that there wasn't even a serious framework for thinking about what this could be. The energy source is chemical. Hydrothermal vents form where seawater percolates through cracks in the ocean floor, gets superheated by magma below, picks up dissolved minerals and gases, including hydrogen sulfide in significant concentrations, and vents back into the overlying water. The temperature of the fluid exiting the vent can exceed 400 degrees Celsius, though it cools rapidly as it mixes with the surrounding seawater. At the base of the vent ecosystem are chemoautotrophic bacteria and archaea, organisms that fix carbon from CO2, not using light energy but using the chemical energy released by oxidizing hydrogen sulfide. The same electron transport logic as aerobic respiration, but substituting hydrogen sulfide as the electron donor instead of organic carbon, and sometimes using nitrate or other compounds as the final electron acceptor instead of oxygen. The process is called chemosynthesis, which is a functional parallel to photosynthesis. Same logic of fixing inorganic carbon into organic compounds, different energy source. The microorganisms doing it are the primary producers of the vent ecosystem. Everything else is eating them or eating something that ate them. The tube worms deserve specific attention because their solution to living in a vent environment is one of the stranger biological arrangements I've encountered in this general area. Riftia pachyptela, the giant tube worm of hydrothermal vents, has no mouth, no functional gut beyond a vestigial structure, no way to eat in any conventional sense. What it has instead is a specialized organ called a trophosome, filling much of its body cavity, packed with chemoautotrophic bacteria in densities that have been estimated at billions of cells per gram of trophosome tissue. The bacteria fix carbon using hydrogen sulfide the worm delivers to them through its bloodstream. The bacteria feed the worm with the organic compounds they produce. The worm is, metabolically, a housing structure for its bacteria. It has specialized hemoglobin, a different configuration than yours, capable of binding both oxygen and hydrogen sulfide simultaneously without being poisoned by the sulfide the way your hemoglobin would be, and it uses this to supply both compounds to the trophosome. The bacteria get their substrates delivered. The worm gets its nutrition from bacterial production. Neither organism functions without the other. I've never seen a live Riftia. I've seen preserved specimens in a collection, which is a deflating experience. They look like wet pink tubes, entirely without the quality that makes them extraordinary, which is the living chemistry happening inside them. The photograph I keep coming back to is from an ROV footage still, a cluster of them around an active vent, white tubes, red plumes extended into the water, surrounded by the chemosynthetic bacterial mats that coat every surface near the vent. The red of the plume is hemoglobin, blood essentially circulating through an external gill structure, picking up sulfide and oxygen from the water. A 2-meter worm in darkness, at crushing depth, feeding on bacterial metabolic output, living in water that would be uninhabitable to almost anything else. The discovery that vent ecosystems existed forced a re-examination of several things simultaneously. The definition of the habitable zone, the range of conditions under which life is possible, expanded significantly. If the absence of sunlight, the presence of toxic sulfide concentrations, high pressure, and extreme temperature gradients were not barriers to complex ecosystems, then the list of environments that might support life, including on other planetary bodies, got longer in ways that were uncomfortable to fully think through. The origin of life question got more complicated and more interesting. Hydrothermal vents, not necessarily the high-temperature black smokers, but a different type, the alkaline vents like those found at the Lost City vent field, have become a serious candidate for the environment in which life first emerged. The chemical gradients, the mineral surfaces, the constant flux of reduced compounds from the Earth's interior, these conditions have been proposed as a plausible setting for the origin of chemosynthetic metabolism, which may have preceded photosynthesis in the evolutionary sequence. Life possibly started in the dark, in chemistry rather than light, and then later found its way to the surface and developed the capacity to use the sun, which would mean photosynthesis wasn't the foundation at all. It was a later innovation built on top of something older and darker and more chemical. The vent ecosystems discovered since 1977, and there are many spread across mid-ocean ridge systems in every ocean basin, have their own distinct community compositions. Different species at different vents, different dominant organisms depending on the chemistry of the vent fluid, the temperature, the depth. Some vent species are related to shallow water organisms. The evolutionary history of how they got to the deep sea, how they adapted to vent conditions, is a research area with enough complexity to sustain careers. What's clear is that the colonization happened multiple times, from multiple surface lineages, and that vent conditions have acted as strong selective pressures, producing convergent solutions in some cases where distantly related organisms have arrived at similar adaptations. The clams at hydrothermal vents have chemototrophic symbionts in their gills, a different solution to the same problem the tube worms solved with the trophosome. Two different animals, two different arrangements. Same basic dependency on bacterial chemistry in the dark. Somewhere at this moment a vent is active on the ocean floor. The superheated fluid is rising. Bacterial mats are coating the mineral surfaces around the vent opening, running chemosynthesis, fixing carbon in the absence of any light. Tube worms, if the vent has been established long enough to allow colonization, are extending their plumes into the water. The ecosystem down there is not aware of the surface. It has no relationship to sunlight. It doesn't need the sky. It runs on the heat of the Earth's interior, on chemical gradients, on the same electron transfer chemistry that life has been using since before oxygen existed. Down there, the planet is enough. There's a particular moment that happens sometimes when you've been thinking about biology for long enough. Not an insight exactly, more like a shift in how the ordinary looks. You're outside or near a window and something, the condensation on a glass, the smell of rain on warm pavement, the sound of wind moving through whatever trees are nearby, briefly stops being background and becomes foreground. The world stops looking like scenery and starts looking like process, like something is happening everywhere, continuously, at scales above and below what you can perceive, and the apparent stillness of it is a limitation of your senses rather than a property of the thing itself. I've had this happen in unremarkable places, a parking lot in Cincinnati after rain, standing next to my car looking at the wet pavement and thinking about the microbial biofilm coating every surface. A highway overpass of all things, thinking about the nitrogen compounds in vehicle exhaust being processed by nitrifying bacteria in the soil below the embankment. The sewage treatment facility, where it happened most intensely if I'm being honest, standing next to a tank of activated sludge, watching the surface and knowing what was in it, the diversity of it, the functional sophistication of a microbial community doing in an engineered tank, what natural systems do without infrastructure. The world keeps being more biologically active than it looks. There is no clean name for what this chapter is trying to describe. The Gaia hypothesis, the idea that the Earth behaves as a self-regulating system, that biological processes maintain conditions suitable for life, has a complicated history. James Lovelock and Lynn Margulis proposed it seriously, it was dismissed, it was partially rehabilitated. It exists now in a softer form that most Earth system scientists find more acceptable than the original strong version. The strong version implied intention, or at least a purposeful homeostasis, that the biology doesn't support. Organisms don't regulate the planet because they're trying to. They regulate it because their collective metabolic activity has physical and chemical consequences that feed back on the conditions for their own survival. The distinction matters. Not because one version is more comforting than the other, but because the true version is, I think, more extraordinary. A self-regulating system that nobody built, that no organism maintains, that emerged from billions of years of metabolic activity and evolutionary selection, and the physics of how gases mix and how heat moves through water. That's a stranger thing than a designed system, stranger and more real. Consider what this series has actually covered. If you've been following from the beginning, cyanobacteria producing oxygen as a metabolic byproduct, transforming an atmosphere. Decomposers cycling carbon and nitrogen through soil, making those elements available for the next generation of organisms. Nitrogen-fixing bacteria pulling atmospheric nitrogen into biological circulation, without which protein synthesis across most of the living world would stall. Phytoplankton drawing CO2 out of surface waters, exporting carbon to the deep ocean when they die and sink. Sulfur reducing bacteria processing organic material in anaerobic sediments, running a cycle that connects ocean chemistry to atmospheric sulfur. Termite colonies and their gut microbiomes processing lignocellulose, returning carbon to the atmosphere as CO2 and methane. Earthworms restructuring soil physical chemistry. Iron chemistry linking dust transport from continental deserts to ocean productivity across thousands of kilometers. None of these organisms are doing any of this for the planet. They are doing it for themselves, for ATP, for carbon, for electrons. The planetary consequences are incidental to every individual metabolic transaction, and yet the aggregate of those transactions across geological time has produced and maintained an atmosphere with a particular composition, oceans with a particular chemistry, soils with particular structures, climate patterns that have stayed within ranges compatible with complex life for hundreds of millions of years. The planet holds itself in a condition suitable for life because life is continuously inadvertently doing the holding. I've been writing about biology long enough that I've developed a reflex. When someone describes a system as stable, I start looking for what's doing the stabilizing work, because stability is almost never passive, it's maintained. Something is running to keep it where it is. In ecosystems, that maintenance is distributed across the metabolic activity of every organism present, each running its own chemistry, each altering its local environment in small ways. the aggregate of those alterations producing conditions that are, in some loose sense, self-correcting. This doesn't mean the system can absorb any perturbation. It means it has been absorbing perturbations, volcanic eruptions, asteroid impacts, glacial cycles, mass extinctions, for billions of years and has so far recovered. The recovery times are geological, the mechanisms are biological. The outcome so far is continued habitability. So far is doing a lot of work in that sentence. I'll leave it there. There's something I've been circling without quite landing on across this entire series, and I want to try to say it directly even if the saying of it is imprecise. The organisms we've talked about, the cyanobacteria, the decomposers, the nitrogen fixers, the iron oxidizers, the phytoplankton, the sulfur cyclers, the termites, the earthworms, the tube worms running on-vent chemistry in the dark. They're not supporting the planet the way a pillar supports a roof. They are the planet's chemistry running biologically. The distinction between the living and non-living parts of the Earth system, at the scale we've been working at, is genuinely blurry. Carbon moving through organisms and carbon moving through rock weathering are part of the same cycle. Nitrogen fixed by bacteria and nitrogen deposited by lightning are both inputs to the same pool. The ocean and the atmosphere exchange gases through physical processes and biological ones simultaneously. Biology is not on top of the Earth's chemistry. It's inside it. It always has been. You breathe tonight in an atmosphere that organisms made and are making, and will continue to make after every individual organism currently alive has died and been processed back into the cycle. The nitrogen in you will leave you eventually, through excretion, through decomposition, through the long chain of transfers that cycles elements through living systems. It will enter soil, enter a plant, enter something that eats the plant. The carbon in your cells will return to the atmosphere as CO2 and be fixed again by photosynthesis into something's organic tissue. You're a temporary configuration of elements that have been cycling through living systems for billions of years. The configuration is temporary, the cycling is not. If this series has meant something to you, if you have found yourself thinking about soil or sulfur or deep ocean vents in the quiet space before sleep, then it has done what I hoped it would do, not made the world simpler, made it more inhabited, more active, more strange in the specific way that real things are strange when you look at them without the filter of familiarity. There are people who've kept this channel going through membership, through super thanks, through simply staying. And I'm aware that gratitude for that can sound formulaic when said directly, so I'll say it sideways. This work exists because enough people found it worth keeping alive, and that's not a small thing. If you've found something here that makes your nights a little more interesting, a like or a subscription is a quiet way of saying so, and it helps more than it probably looks like it does from your end. The cycle continues. It always has. Sleep well.